In 16.2, we are going to look at a little bit more about pH, and we're going to look at what are called titrations. So the first thing, a little refresher, we've worked with and we've looked and talked about acid-base indicators, but this is just going to be a substance where the color will change depending on the pH. And typically these are going to be weak acids or bases that are used to make these indicators. And this chart shows you several different indicators. And so one that we will use is phenolphthalein. In more acidic solutions, it's going to be clear. And then as it turns to a base, it will be pink. Phenol red, it's going to be yellow and pink. And then here's bromethyl blue is one that we have. There's blue, and it's going to change from yellow to blue. And then around 3 or 4 is where you're going to get that change. It's going to be a green color. So there are many different types of acid-base indicators. We looked at cabbage juice. We used pH paper. So you should be pretty familiar with this at this point. To make indicators, they're going to often use a weak acid, and the anion part of your indicator is going to be attached to a hydrogen, and that's what's going to cause the different color change. And so the hydrogen in the indicator and the indicator ion would be different colors. Transition interval is that pH level where you get the change. And this is different. You can see different for different indicators. So for phenolphthalein, when the substance is changing anywhere from 8 to about 10 is where you're going to get that change. If we've got bromethyl blue, the changes between 3 and 5. So this area that you can see where your color's changing, that's called the transition interval. Universal indicators are going to be made up from several different indicators put together. And so we used this in class where we had, it was a liquid that we were dropping into our solutions. And so for each different pH level, you're going to get a different color. pH meters are something that you could use, and you would just simply place these in your solution, and it's going to tell you the pH by using the voltage that is carried between the electrodes. And so the more of an electrolyte the solution, the more of an acid it's going to be. Titration is an experiment we're going to look at in this section. And so this is where you're going to have a solution that you know the concentration and another solution that you do not know the concentration. And you're going to be able to determine the concentration of the unknown by mixing the two substances. So basically, this is what it's going to look like. You're going to have an acid and a base, and so one of those will be placed in the flask, one will be in the Biuret tube. And then you will use an indicator, and in this example, and what we'll use in class, is going to be phenolphthalein. So here, they're separate. Let's say the acid's in the flask and the base is in the Biuret tube. And then they're slowly going to drop some base down into the acid. And they will continue to add that until we get a slight color change, and that would be our ending point. If the color gets too dark, we've put too much in. And so we will be able to measure the volume of the substance in the flask with the volume of the substance that we've added, and that will help us determine the concentration of the unknown. The equivalence point is where the two chemicals are in chemically equal amounts. And the end point is where the color change would occur. And so you would have to pick out an indicator to use depending on what acids and bases you're working with and where they're going to have that color change occur. Typically, if you're going to be using a strong acid and a weak base, you're going to have an indicator that's going to change color with a pH lower than 7. If you have a weak acid and a strong base, you want your pH to change higher than 7 for your indicator. And if you have weak acids and weak bases, this will not work. And then a standard solution is the solution that you know the concentration. And so you have to know the concentration of one of the solutions to be able to solve for the concentration of the unknown. A primary standard is going to be something that's very purified so that you can check for your accuracy. So you will first have to write a balanced equation for the substances that you're trying to work with. You're going to determine what their chemically equivalent amounts would be, solve for the moles of your solute or the unknown, and then you would be able to determine the molarity of your unknown.